Then first speaker of the session is Jacob Miller. He is a researcher in the Yale University. Then title of his talk is Toward the Cognitive Global Structure and Functional Architecture in Prefrontal Cortex. So please welcome Jacob.
Thank you so much, Nicola and Hiromasa, for having me. I'm excited to talk about this. I'm looking forward to any questions after, so please, after this, at any point of the day, I'm happy to chat about any of this, and I'll try to keep this a little more casual and fun because it is an educational session. So today I'll be talking about trying to map a cognitive globe of structural and functional architectures in the prefrontal cortex. So if we were having this meeting 100 years ago, we might not be talking much about the prefrontal cortex because even less than 100 years ago, Forster and Penfield and others called this area of kind of anterior PFC silent or uncommitted cortex. So it was not that long ago in human history that we didn't think this part of the brain really did much that was important for human behavior. Now, of course, we know that's incorrect. So if you look at people that have damage to areas of the lateral prefrontal cortex, which you can roughly divide into dorsolateral, rostral, and ventrolateral, people have all sorts of cognitive deficits, ranging from things like problem solving, planning, working memory, motivation, and attentional issues. And there's a real kind of cognitive porridge, you could call it, of issues that happen when you have damage or tissue loss in this area of the brain. And so the question I'm asking today, and I'll try to get at, is what is the organization of the human prefrontal cortex? And one idea of this, that there's so many deficits when you have these kind of lesions to the prefrontal cortex and that it's involved in such a wide array of cognition, is that maybe there really isn't something like a fundamental organization. Maybe the prefrontal cortex is sort of everything everywhere all at once. So every region, if there even are different regions in this area of the brain, are sort of all contributing equally and doing about the same thing here. And while that does explain a lot of data, today I'll try to put forth a perspective that instead we could kind of think of the prefrontal cortex as operating as a sort of cognitive globe, where there are different regions along the lateral prefrontal cortex that have different anatomical properties and related but somewhat different functions depending on their role in a given task or across wide arrays of cognition. And the reason this matters, as Nicola was saying, is not just for mapping's sake, but rather to get at function itself. So I'll reference this a few times later in the talk, but the reason this is important, I contend, is that the prefrontal cortex is really important in serving as a sort of integrative learning system across multiple different areas of the brain with strong inputs and projections back to sensory streams, MTL, and long-term memory systems that helps us navigate goal-directed behavior. So today I'll hope you'll take away 3 things from this that I'll talk about. First, I'll show you that individual variability in the prefrontal cortex is meaningful for functional organization. I'll then show you consistent data that there's a mesoscale organization of functioning in the PFC along a rostral-caudal axis. And then finally, we'll take a fun dive below the level of neuroimaging voxels to see that there is anatomical functional coupling in this area of the brain across scales ranging from millimeters all the way down to scales of microns. Okay, so first, we're going to talk about individual variability in prefrontal cortex functioning. And this is a really hard problem. And the reason that is because there's a lot of mess going on in this area. So if you look at, for example, analyses of different resting state functional networks, a ton of them converge within individuals in the lateral prefrontal cortex, and they're overlapping within the same people. There's a whole host of 10 to 20 or more cytoarchitectonic areas in the lateral prefrontal cortex that are all in very close proximity with debated boundaries and areas to each other. And if you look at electrical stimulation and tracing data, the lateral prefrontal cortex has connections to sort of maps of different areas of the brains that are all overlapping with each other. So this is a pretty daunting challenge, and hopefully we'll take a little bite out of it today and try to get somewhere. Another reason why this has been so tricky is that even great anatomists of the past have sort of all come up with different names, structures, and subdivisions of their favorite drawings of the prefrontal cortex. So if you look at simple sort of cylindrical folding labels of structures in the prefrontal cortex, depending on your favorite anatomist of the past, they might all have many different names, divisions, labels or not, for the same structures within the lateral prefrontal cortex. And what that's led to in modern neuroimaging is that when you look at individual subjects, it's a lot different than what you see on template brains. So for example, if you average cortical surfaces together, here is a left hemisphere cortical surface showing sulci indentations in dark gray, that as you average more and more brains together, these sort of individually meaningful indentations almost disappear entirely, whereas you can see they're very prominently labeled in white within individual subjects. And these structures that are lost in group averages are called tertiary sulci. And the reason I'll contend they're important is that they have an extended developmental trajectory. They're the last cortical folds to form in utero. And they can be labeled within individuals. And I'll show you that they serve as functional landmarks as well. So briefly, I'll point you to some work that we did that we set out from a mapping that Petrides had done in templates to say, can we identify these individual structures within individual subjects from the Human Connectome project? We followed Petrides labeling to show that instead of averaging your data, if you take a look at individual subjects, there are really prominent tertiary sulci cortical indentations within any individual you look at in this area of prefrontal cortex. And this is the case if you look at postmortem brains, sections of postmortem brains, cortical surfaces from MRI. Any way you look at this, you're going to find these indentations within individuals. So why does this matter? Well, it matters because it can help us get at this functional mapping. So for example, identifying these set of structures, what I'll call the PMFS, or a tertiary sulcus in the lateral prefrontal cortex, it can identify, just by labeling the anatomical landmarks, areas with different network functional connectivity profiles, and even changes in microstructure like gradients of gray matter myelination across cortex. These sulcal indentations have different microstructure across cortical depths, even compared to areas right next to them that are gyri and not sulci. And so the really cool thing about kind of embracing this variability in anatomy here is that we can find structures that are prominent within individuals that have a variable location and a shallow depth, but that serve as landmarks for distinct functional network profiles and even for microstructural gradients. And this is new work, but it's a very old idea. So even if you go back to the Voets or Friedrich Sinides and the great German neuroanatomists of the past, you can see that Sinides here on the bottom left hypothesized that the tertiary sulci would serve as these sort of transitional zones between functional and anatomical areas. And so if you take a step back and look at a bunch of different kinds of data, be it multimodal parcellations, lesion data, functional activations, these individually specific sulcal indentations demarcate a lot of really interesting findings that we may all have in our data. And again, can we even take this further to show why does this even matter, even if you have a structure-function relationship? If you look at data we haven't labeled, but we teamed up with Sylvia Bunge at Berkeley to look at developmental trajectories of cognition, variation in the morphology of these structures predicted cognitive development across adolescence. And what I'm promoting today is not that everybody in their own data manually identifies and learns how to identify excruciatingly these structures within individuals, but rather that we can use this work to build tools so that you can keep your data in its own native space and identify these structures in totally unseen individuals. And so here's some work that does that with a little bit fancier machine learning on our manual anatomical labeling. Okay, so this individual variability in the prefrontal cortex is meaningful. And I'll also tell you now that there's large-scale functional gradients in the prefrontal cortex. So the prefrontal cortex is often thought of, and this is my own bias as a working memory researcher, as being really critical for serving functions in working memory. So here the idea is that when you don't have access to a sensory stimulus anymore, there's persistent activity in the prefrontal cortex that maintains things we want to remember. But it's really interesting if you look at human neuroimaging versus monkey electrophysiology literature, you come away with really different ideas of what the prefrontal cortex does. So for example, classic findings in monkey prefrontal cortex show that there are neurons and groups of neurons that maintain memories of something you can't see anymore. Whereas in humans, the prefrontal cortex functioning seems to be more about general high or low levels of activity associated with things like cognitive control or attention. And this is interesting. This matters to us as functional neuroanatomists because it brings up a lot of questions. Is this something of a functional difference between these areas? Are they really different anatomical areas? Is it a species difference or a cognitive difference? And one difference here, aside from the kind of recording method itself, is that humans and animals come at cognitive tasks from a very different standpoint. So as neuroimagers, a lot of us might collect data from a naive participant 5 minutes after we've explained to them the task and throw them in the scanner, whereas a graduate student doing monkey electrophysiology might spend 2 years training an animal to do a task and then record from neurons in the prefrontal cortex. And so in one study I'll briefly tell you about, we tried to flip this on its head and say, well, what if we train humans with really long-term repeated learning? Can we see something that looks more like the findings you see in monkey prefrontal cortex? And so to do that, we took fractal stimuli that were naive to subjects that have been used in classic kind of hippocampal pair coding studies before. We generated a bunch of them and gave them to naive participants and scanned them for months and months and looked at their PFC activity. Because this was a very demanding and fun study that involved over 20 sessions of collection for each participant, these were not random participants, but in fact myself and 2 postdocs in Mark Desposito's lab, and there's us having the data collected on ourselves there. So what we did was complete 24 functional MRI sessions each across 3 months. We each had our own group of these unique fractals, and we completed a working memory task using these and other similar stimuli which changed each session. I won't go too much into the details here, but I'll tell you the full data set's available as well on Open Neuro, and we collected massive amounts of data for each subject, including upwards of 5 hours of resting state data total for each person, and over 1,600 trials each of working memory data in the functional MRI setting. So we did this in order to show potentially an effect that we learned. So for example, we get better at this task over time for stimuli that we've seen over and over again. Again, I won't go into the exact kind of details of the task and setup here, but all this is to say is that it sets us up to then test this question about neural functioning and structures in the prefrontal cortex, which is, across long-term learning, do patterns of activity in the human brain come to look something more like what you see in non-human primate electrophysiology? So that is there something we can detect with bold fMRI after months and months of learning that looks like something that you'd think would only be detectable with neurons in the prefrontal cortex? To do this, in brief, we used a pattern similarity approach. So we took the activity on every trial of seeing each of these stimuli and remembering them, and calculated pattern similarity across all trial conditions. And we could do this across every session of the task. And briefly, I won't go over the specific methods, but this allows us to then ask, how does the representation in the prefrontal cortex or other areas change across these months of repeated learning? And so then what it can allow us to do is test for specific kinds of representations in the data. And what we found was something really interesting. So in the ventral midlateral prefrontal cortex that's shown in light green on the bottom middle there, we found that specific activity patterns were associated with individual items in the prefrontal cortex across the course of months. And this is something that'd be much more common to find in a non-human primate electrophysiology study than a human fMRI study. On the other hand, in more caudal regions of the prefrontal cortex, in purple and yellow, what we see is not item-specific representations, but categories of stimuli. So also baked into this task were that stimuli belong to different categories that we learned over time. And so what we've shown here is that there's somewhat of a gradient of representations moving from item-specific to categorical representations across the rostral-caudal axis of the prefrontal cortex. So by doing this really intensive cognitive training and kind of complicated experiment, we can then kind of come up for air and see this large-scale organization emerge in the prefrontal cortex, but only across months of learning. And this corresponds really nicely to these classic studies of non-human primate electrophysiology in prefrontal cortex. So here we're finding something analogous in a cytoarchitectonically similar region of something like ventral area 946 in the prefrontal cortex, which is that we find representations for individual memory items you're holding in mind. And this is summarized here in the paper. You can see we made this summary figure which shows a whole host of learning effects across this gradient of rostral caudal organization in the lateral prefrontal cortex. So there's this really interesting sort of millimeter mesoscale organization across this swath of gray matter in human prefrontal cortex. And this matches really nicely to other kinds of data people have found about this organization. So for example, even doing electrophysiological recordings, there's data from several labs, including Christos Constantinides and Earl Miller's lab, showing that if you put Utah rays or electrodes across monkey prefrontal cortex, you can find also analogous different kinds of representations and activity profiles along the swath of cortex. So there really seems to be an interesting correspondence between kind of large-scale anatomical areas and functional representations in the prefrontal cortex. And this is supported in general by a whole host of other evidence for this, which I think is really interesting because it points to kind of a multimodal convergence, that there's some sort of structural functional relationship here in these areas. So for example, if you look at track tracing data, there's interesting gradients of input and output connectivity differences across prefrontal areas. You see the same thing if you look at large studies of human neuroimaging and different cognitive control processes. You can even see this in lesion data. So for example, both in experimental lesions in primates and in damage in human stroke patients, you can see that damaging anterior versus posterior areas of prefrontal cortex shows different functional representations and cognitive impairments emerge. And even large-scale recordings in monkey PFC show this kind of general organization. So even something as simple as anatomically thinking about macro-scale landmarks across human lateral PFC can tell you something interesting about the functional and cognitive implications of what you're looking at in prefrontal cortex. Okay, but where does that leave us all as neuroimagers? So there's a mesoscale organization of functioning. That would be great if that's the only scale of functioning in prefrontal cortex, if everything happened at the level of millimeters or centimeters and we could solve everything by doing functional MRI. Unfortunately, that's not the case, and neurons are in fact very small. And we're left with this problem of thinking about what happens at circuits below the level of voxels. And what I'll hope to convince you of in the last 10 minutes is that understanding what's in a voxel in your area of interest can then help you better understand what kinds of studies and what kinds of approaches you can take, especially from the context of prefrontal cortex. And why this matters, again, to go back to this recent review about prefrontal cortex learning, is that there are analogous findings between the human fMRI and monkey ePhys, but that a lot of the stuff we really want to get at with our neuroimaging data, like neural dimensionality or representational geometry or kind of fancy neuron level analyses, we really need to, in order to do that, think about what the correspondences are in our data. And to represent the more skeptical electrophysiologists that might not be in this room, what they would typically say is that, well, that's just impossible with functional MRI. So for example, if you look at recordings from an area like V4 in posterior visual cortex, these diagrams just show briefly that across a Utah array, you can generally find that channels you're recording from have more similar functional responses at the level of millimeters. Whereas if you look at an area in caudal prefrontal cortex, like area 8A, there might be an individual neuron in there which has a tuning for a direction in space totally different than its neighbors. And so kind of a classic argument for the importance of electrophysiology is that, well, you'll never be able to detect those neurons with functional MRI, so the only gold standard is recording those individual neurons with electrodes. But I'm going to tell you here that maybe we can do more than that with neuroimaging, and the reason is that we can take advantage of the kind of mid-micro scale organization in different areas. So if you think back to kind of basic neuroanatomy, we learn when we first learn neuroscience is that there are these really beautiful orientation columns in an area like V1, where, for example, if you inject a tracer into a neuron in layer 3 of primary visual cortex on the right, its neighbors will all show that it's connected in a cortical column, and they generally prefer the same location in space. In prefrontal cortex, what you find is a lot different. So instead of something like a family right there by that neuron, if you inject that neuron, there might be sort of a mini column of neurons adjacent to it, but then you can see that it kind of skips over to neurons hundreds of microns away in this sort of mini column pattern where it skips gaps between there. And so the functional organization of PFC might be actually a lot different than what you see here. And so there's questions about, well, for one, even is there a microscale anatomical functional correspondence, and where does this leave us as neuroimagers in terms of how we can try to understand these effects of selectivity and tuning and function if we're stuck with a voxel that's maybe 1 millimeter or 2 millimeters big? Well, there's been recent work that kind of helps us understand the bottom left here, which is what is the scale of functioning and anatomical correspondence in prefrontal cortex below the level of millimeters. So one study comes from Liping Wang's lab in China, where in a science paper in 2022, they did calcium imaging in prefrontal cortex and showed some correspondence here. And I've looked at some of that data, and you can see that if you just look at how close neurons are compared to how similarly they are tuned, how similar their function is, it's really strong in V1, like that red line that starts off really high and then drops down. Whereas in PFC, it's much flatter, but it's still above 0. So while there isn't as beautiful of a structural functional correspondence as V1, it's perhaps beautiful in a different way, but there is some organization there. And there have been several other studies that have come out very recently which show this point. So for example, if you look at recordings in monkey prefrontal cortex and you pick 2 different areas of electrodes or channels, and you just ask the simple question here on the top right of, are neurons that are closer together more likely to show similar functional activity profiles? The answer in prefrontal cortex seems to be yes, but not as much as visual areas. But the fact that it's yes and even holds at a distance on the bottom left here, you can see even at 1 to 2 millimeters, there's still a correlation between neurons and prefrontal cortex. So maybe there is some sort of hope for us as functional neuroimagers to study kind of cross-scale functioning in prefrontal cortex. Of course, that's sort of belying the real complexity of the brain, so I'll leave you with this picture from Jeff Lichtman's lab at Harvard, which shows in just a 1 millimeter cubed section of human temporal cortex, there are upwards of 60,000 neurons or cells and 150 million synapses. So I'm not saying that we can detect that with neuroimaging, but what I'm saying is that there's more there than perhaps meets the eye functionally. And why is that? So the note I want to leave on is that we can't just think of neuroimaging as averages of averages, but that we have to really understand what's happening below the level of voxels and think about it in order to better interpret our data and design studies. So for example, there are proposals from Nico Kriegesgård and others that if voxels were just doing a simple mean average of everything around it, that would not really explain a lot of the findings you see in functional MRI. And instead, the sort of complicated organization of arterioles and veins in the blood supply of the human brain actually could be an advantage in terms of the fact that voxels sense things differently and could be sensitive to different groups of neurons within voxels than just being a simple average. And this has been shown computationally by Brad Love's lab, where they show that kind of if the brain just worked in sort of hard boundaries, like on the right of this figure, if you average things with fMRI, you wouldn't really find much. But because of this correlation between neurons in this sort of smoothness here, you do have a chance with fMRI to actually find and pick up on this functioning even at the level of millimeters. And so what I want to leave you with is that with functional MRI, it's harder than in visual areas, but it's not impossible to detect the kinds of interesting stimulus and cognitive information we want to study in the prefrontal cortex at scale. And so I'll leave you with a few bits of sort of pedantic advice for human neuroimaging if you're interested in prefrontal cortex functioning and structures. So the first I would say is to avoid excess averaging, especially across subjects, and that really that what seems like something that makes the problem harder can actually make it easier. So that is, embracing anatomical variability can actually help us boost our understanding of structure, functioning, and behavior in prefrontal cortex. And also that voxels in different areas of the brain, while they're the same shape when we acquire functional MRI data, what drives those voxels across areas may be really different. So interpreting a pattern of activity or decoding or something of voxels in V1 versus prefrontal cortex is really different, and you have to think about what might be driving those voxels and why across areas. And finally, that we can leverage these insights to make better methodological and experimental design choices. So one is that experimentally, as long as we hold SNR high, which is a challenge, going down to voxels like 1 or 1.5 millimeters is going to be better. We need more trials within subjects, so the more data you have, the more interesting and demanding your design is with complex stimuli, the more likely you're going to find these effects in the prefrontal cortex, and then we need to embrace that variability of activity instead of kind of average it away. And finally, with our data in PFC, avoiding spatial smoothing, considering the level of individual voxels or the many 10 to 20 subregions of the lateral prefrontal cortex is going to help you kind of ride this wave of variability and get more out of your analyses. So if you're interested in these questions and more, I'll leave you with a few texts I think really go into all of these aspects more. The top one is a meeting which took place a year or two ago, which was the Ernst Strungman Prefrontal Cortex Great Meeting of the Minds, which was edited by Marie Banich, Suzanne Haber, and Trevor Robbins. And so it's a chapter of many of the people in neuroscience, OHBM, and others, writing down everything they know about the prefrontal cortex, along with some other kind of fundamental reviews, which are kind of nice starting points into understanding these questions. So again, what I hope to leave you with here is that individual variability in prefrontal cortex anatomy is meaningful. There's a mesoscale organization of functioning across the human lateral prefrontal cortex, and that there's an anatomical functional coupling across scales from microns all the way to millimeters. And finally, I'll plug that I have recently started a lab at the University of Miami. So if you like beaches that are much, much warmer than they are right now in Brisbane, please reach out if you're interested in any of these questions or more. I'm happy to take any questions now and chat after. So thank you so much.
Thank you very much. So, okay, we have some time for question discussion. So, okay, please.
Very nice talk. I liked your recommendations on your last slide of things that researchers should do.
Yeah.
One thing that I didn't notice, unless I missed it, was recommendation to do something like hyperalignment. And I just wanted to hear your thoughts on that.
Yeah, it's a good question. I think it's hard in PFC because I think the premise, from what I understand of hyperalignment, sort of relies on having a nicely defined functional area. So for example, something like perihippocampo place area or one of the emphuse face areas, which is great if you have that. The problem when you get to, I think, really hierarchically complex cognitive control regions is what is a functional area, right? Sort of what is there's no sort of one localizer you could do. You could do 100 tasks and they all might light up the prefrontal cortex and a broad swath of it. So I think in this case, it really almost even matters more than in something like ventral temporal to kind of leave that anatomy unmorphed in some ways, yeah.
Next question? Yes, please.
Hey, thank you. Great talk. I wanted to comment on or dig into your question or your proposition that variability in a prefrontal cortex kind of matters, individual variability. So, because we found individual variability in V2 and V3 in humans. That's not idiosyncratic, but they're patterns that different people share. But the question that we have is it meaningful? Because, yeah, it's not idiosyncratic, so it's reliable, but that doesn't mean it's meaningful. So the question, meaningful would mean it has an effect on perception, for example, or cognition. And I just kind of thought, what are your thoughts on that?
Yeah, it's a really good question. I think that the word meaning is very loaded, and we all might come away with what is different from that. I think, and I certainly didn't mean to say that there's not interesting and perhaps meaningful variability in other areas of cortex and structures. There are everywhere. I think, to me, as a cognitive neuroscientist, the meaning of individual differences in those kind of regions will ultimately all depend on a link to behavior. So that's, I think, from a cognitive neuroscience perspective, you can show in your data that in individual tasks or across tasks that there's a reliable association to behavioral differences that track anatomical functional correspondence. That, to me, would be how you get that meaning. And there are, of course, a lot of debates about whether the variability is a spectrum or if there's different subgroups and stuff. And I'm happy to talk more about kind of the debates about whether it's really subgroups or everyone's different or something like that. Yeah. Thank you.
Hello. Didn't quite understand your contrast between V1 and frontal areas, because in both areas you have this patchwork of intrinsic connections that skip. To me, the biggest difference between these areas is more that the periodicity of that was shown by Jennifer Lund and Nishiro Fujita and others, that the intrinsic connections become further and further away in scaling with the size of the pyramidal cells. So my thinking of that is that if you can find the correct parameter, what is actually being mapped in the map of that area, it should make it easier rather than more difficult to find because the modules are larger. Would you comment on that?
Yeah, I think it depends. I think I generally agree with the premise that, yeah, there's something we can take out of the tracing data that will make it easier, and why that is tricky to understand. I think one of the harder parts of PFC, and I was, of course, simplifying a lot of that data, is that it's not just that there are skips and sort of that periodicity, which is different than V1, so I fully agree with that, but that it's also overlapping. So I showed that image of sort of the PFC showing kind of overlapping connectivity to these different large swaths of cortex, so that you might have some periodicity of these mini columns connected to each other, but then a different set of mini columns right next to that, which might have different selectivity that are also periodic. And so that, I think, is the really tricky challenge. And I think a good demonstration of that, sort of at the more mesoscale, is this paper from Zhu and Bob Desimone from Neuron in 2022, where they stimulate a bunch of different areas of the PFC and show there are overlapping but distinct connectivity to a bunch of different areas of the brain. And I'm happy to chat more about kind of like what might be there or not. Yeah.
So last question.
Okay. Hey, Jakob, nice talk. So I have a simple question. So, you mentioned a tertiary subsite as a landmark for the PFC subregion. So during the function fMRI gradient data, do you see any relationship with these gradients in those specific
Yeah, it's a good question.
not?
So in that 2021 paper, I didn't show it, but we look at sort of large-scale meta-analytic activations to show that there's kind of rostral-caudal differences in activity, and there's been lots of papers that show that. And then even with individual tasks, there's also lots of work on sort of some signal and some noise about what's different between gyri and sulci in terms of activations and blood flow and stuff that affects functional MRI. Some of that being interesting, some of that being sort of an effect of magnetic fields and stuff like that. But in general, yes, those focal landmarks also show functional activation differences, especially across like batteries of tasks. Yeah.
That's great. Thank you.
Yeah.
Sorry, for the interest of time, we have to move on, but thank you again for Jakob. Yes.
Thank you.